Hi, everyone. In this podcast, we're going to talk about what Crick called the central dogma. And basically, what we're going to talk about is how our genetic information flows from DNA to RNA to a protein, and then to add something Crick didn't really discuss, to a phenotype. So let's get started. Okay, I love this slide because it's a really quick overview. So you know that transcription is the process where we take DNA and we copy it into RNA. And we're specifically going to talk about mRNA, which stands for messenger RNA. And mRNA's job is to leave the nucleus, head out to a ribosome, and then be copied into a polypeptide. So what's the difference between polypeptide and a protein? A polypeptide is a long chain of amino acids. It hasn't been edited or folded. Once those things happen, then it's officially a protein. Just a quick refresher from genetics. You guys know that DNA and RNA are collectively called nucleic acids, uses complementary base pairing. So adenine would always base pair to thymine with weak hydrogen bonds, and cytosine would always base pair to guanine. So if I have a DNA that says TAC, this is my DNA, which you already kind of knew because I had a thymine in there, not a uracil, and I'm going to copy this into mRNA, then I know I would use the complementary base pairs. So T would form hydrogen bonds with A would form hydrogen bonds with U, and C would form hydrogen bonds with G. Okay? So why do we use a triplet code? And by the way, this triplet code is what we call a codon. So a codon is found in DNA or mRNA. Usually when we discuss a codon, we are speaking about mRNA. So why do we have to have 3 bases? Well, I have 20 amino acids. Okay, and I have 4 bases. I have A, T, C, and G. Clearly, I can't have one base code for one amino acid. I only have 4 bases. I have 20 amino acids. Well, that's not going to work. Okay, what if I did 2 bases? So I just needed A and T to code for amino acid. Well, 4 to the second power is 16, so that doesn't work either. I need 20 different codes for my 20 different amino acids. And this is how we get to our codon. 4 to the third power is 64. All right, well, that's way past my 20. This is where we get into the repetitiveness of our genetic code. All right, if you look at alanine, for example, we have 4 different codons for alanine: ACU, ACC, ACA, ACG. That's repetitive. Then we have other codons that don't code for an amino acid, and we call those stop codons. And we'll talk about that a little bit more in translation, but basically that means there's going to be no tRNA that will recognize that, no amino acid. It's going to go ahead and stop the formation of a polypeptide. Okay, so let's go ahead and talk about transcription. So obviously transcription is going to happen in the nucleus because this is where we're taking our DNA and we're copying it into an mRNA molecule. And we're going to use our enzyme RNA polymerase for this. This should sound a little familiar. Remember DNA polymerase for DNA replication? Well, RNA polymerase takes DNA and copies it into mRNA. Let's go ahead and dive into transcription. Transcription is made up of 3 different phases. The first one is initiation. That big enzyme, RNA polymerase, is going to attach to a promoter. The DNA strand will be unwound and we'll have a template strand. This will be the strand that's copied into an mRNA molecule. Okay, so then our next stage is elongation, and this is where we're adding bases to our mRNA molecule using that DNA as a template. All right, the mRNA will be made in the, I'm going to switch colors here, in the 5' to to 3' direction, just like in DNA, because we have to have that 3' carbon open to add our next nucleotide, and we will use complementary base pairing as we discussed earlier. All right, termination is our last stage, and the RNA synthesis ends when we hit a terminator sequence. Now we have this strand of mRNA. When I talk about the initiation of transcription, I'm really kind of shorthanding a little bit of a complex process. But you might hear the word TATA-box. And basically, a TATA-box is a portion of a promoter in a eukaryotic sequence. So you are eukaryotic. You have membrane-bound organelles. Okay. So what is a TATA-box? It's really basic. It has a lot of thymines and adenines, and this is going to be part of your promoter. But that's not it. We also have some transcription factors, and these are proteins, and these are going to bind to the DNA before RNA polymerase can bind as well. We'll have additional transcription factors that will make sure RNA polymerase starts exactly where it needs to go. And once this enzyme attaches, we'll go ahead and add in our bases in a complementary fashion. Now remember, there's no thymine in RNA. So if you have an A in your DNA, then you will have a U in your RNA, uracil. Okay, we made our mRNA, but it's not quite ready to leave the nucleus and head out to the ribosome yet. We got to process it first. Okay, the first thing we're going to do when we begin to process our mRNA is we're going to add a 5' cap and a poly A tail. The 5' cap is a guanine with some phosphates, and the poly A tail is different adenines. They could be just a few adenines or many adenines. So what do these two things do? Well, the first thing they do is they help get it out of the nuclear pores so that the mRNA can leave the nucleus and head out to a ribosome. The other thing they do is once this mRNA leaves the nucleus, it's exposed to being degraded, which that's important. If we have mRNA just floating around in the cell, these genes could be made over and over and over again, which could cause terrible diseases like cancer. But it's also waste if we need a protein quite a bit, like a digestive enzyme. We use those constantly to keep making mRNAs to then make into a protein. So sometimes we want to copy that mRNA more than once. Well, the poly A tail will help us with that. So the poly A tail will protect this protein from being degraded once it's out in the cytoplasm, so it can be copied more than once. But let's say this is a protein that's going to be an important part of like initiating cell division, then we want this to get copied once and be done. Because if this gets copied more and more, that's a lot of cell division, that's cancer. So this kind of decides how quickly an mRNA will get degraded. This also helps the mRNA dock at the ribosome. These will not get translated into a polypeptide at all. The other thing we need to do is cut out our introns and then splice together our exons. I always thought these names were a little bit backwards, but the way I remember it is exons are expressed. And what I mean by expressed is they're made into polypeptides, which become proteins, which then become phenotypes. We're expressing it. And introns go into the garbage. That's kind of not really what happens, but it helps me remember it. So we're going to cut out our introns using spliceosomes, and then we're going to piece together our exons, making this the coding segment of mRNA. Okay, we're ready now to ship this out of the nucleus and head on out to the ribosome. Okay, so what's the spliceosome? The spliceosome is made up of SNRPs. And besides being fun to say, SNRP stands for small nuclear RNA proteins. Okay, those are going to attach to the introns, and when all of the SNRPs get together with about 80 other proteins, they make up the spliceosome. So SNRPs plus proteins make a spliceosome, and basically they're going to bend that pre-mRNA, that's what we call it before we cut out our introns, until those introns snap out, they'll splice together the exons, and we're good to go. All right, now we're ready for translation. So our mRNA has left the nucleus, it's headed to a ribosome, and now for translation, we're going to go from RNA to a polypeptide. Okay, before we get into the nitty-gritty of translation, we have to meet tRNA. So tRNA stands for transfer RNA, and its job is to transfer amino acids. It carries them like this. tRNA looks double-stranded. It isn't. It's single-stranded. It's just looped back onto itself. So tRNA is carrying around its amino acids. How does it know what amino acids to drop off when? Well, it knows because it has an anticodon. And I always remember that tRNA has an anticodon because of this T. Remember who has the codon? mRNA has the codon. So if the codon for mRNA is AUG, then tRNA would have UAC. They'll form a complementary base pair, and that's how tRNA will know to drop off its amino acid. Ribosomes are made up of two different things. They're made up of rRNA, that R stands for ribosomal, and proteins. They're also made up of two subunits, a small subunit and a large subunit. These subunits come together once it's time to translate a polypeptide. The small subunit is where the mRNA is going to bind. Then a tRNA will bind to the initiating codon on mRNA, which is always going to be AUG. Okay, the tRNA will bind here. Once that happens, the large subunit will then come and settle onto the small subunit, and we're ready to go. We're ready to make a polypeptide. Okay, so we're going to look at this polypeptide kind of mid-creation here to see how this works. So my mRNA has already bound to my small subunit. We always have our same start codon, all right? We'll code for methionine. Why is that always a start codon? Nobody really knows. Maybe that's something you can discover. We just think that was kind of the evolutionary default, all right? So the mRNA attached to that small ribosomal subunit, then that first tRNA attached, carrying methionine, and then the large subunit attached. And we started to make our polypeptide. All right, so here's what happens. A tRNA will land into what's called the A site of the ribosome. Once it docks, a peptide bond will form between the two amino acids, which means this tRNA has let go of its peptide. Once it's let go, it goes ahead and it leaves the ribosome. It's going to go pick up another amino acid. And this tRNA will then move to the P site. Once it moves to the P site, the A site is open. And we're just going to start the whole process over again. The tRNA will move and dock at the A site. The two amino acids will form a peptide bond, which means this one's let go of its amino acid. It will go ahead and leave, which means now this tRNA can move into the A site, leaving the A site open, and so on and so forth until we hit a stop codon. Why does the stop codon stop the creation of a polypeptide? Well, we talked about this. It's because there's no amino acid, there's no tRNA that would match that codon. There's no anticodon. So therefore, the ribosomal subunits let go, the mRNA is then released, and it can then be edited into a functioning protein. Okay, let's talk about mutations. So mutations is a change in DNA. And obviously, following that central dogma, if there's a change in DNA, then there's a change in RNA, which means there could possibly, not always, be a change in a polypeptide, which could possibly then change our protein, which could then possibly change our phenotype. Probably the most used example of mutations is the mutation that causes sickle cell anemia. So here we're looking at the codon cytosine thymine thymine. This is our DNA. Mutations occur in the DNA. A point mutation, which causes this thymine to become an adenine, results in the chain event of the mRNA being changed and then the amino acids being changed. So in this case, we're going from glutamic acid to valine. All right, and this is kind of a big deal. Valine is hydrophobic. Glutamic acid is hydrophilic. And you can imagine that hemoglobin, which is, in the blood, and you know how aqueous our blood is, that will completely change the shape of the protein, making it function incorrectly. Okay, I stole this picture from the internet just to do a real quick rundown of mutations. So again, changes in our DNA that may or may not cause a change in our protein. How is that possible? Well, if we're looking at just point mutations, that means we're just changing a base pair. If it's a silent mutation, what happens is we change the DNA. It'll cause a change in our mRNA, but remember how repetitive our mRNA codes were for amino acids? Sometimes there was 4 or more codons that would code for the same amino acid. Well, if that's the case, if this mutation codes for the same amino acid, then we call it silent. Nonsense mutations are when the point mutation causes a stop codon to be coded for when it wasn't supposed to be. So this will end the protein early and you'll have a completely non-functioning protein. A missense mutation means that we are going to go ahead and change that amino acid. And this could be not a big deal if you're changing like a hydrophobic amino acid for a hydrophobic amino acid. The protein may bend the same and it really may not matter. Or this could be horrible. So if you change the amino acid and it has a difference in basic properties, then you could be in trouble. We can do things like add a base. Okay, we can substitute a base or we can delete a base. And all of these are point mutations. Within point mutations, there's a big difference between a base substitution, a base deletion, or addition. In substitutions, we're just changing this one amino acid. That can have a devastating effect or it cannot be that big of a deal. In base deletions or additions, you're changing from that point of the mutation on because you're shifting the reading frame. So this is an example of a deletion. We pulled this U out. So we've completely altered how the mRNA will be read from this point on. This is my new codon. This is my new codon. This is my new codon. So anytime we have a base deletion or addition, we call these frame shift mutations because from the point of mutation on, all the amino acids will be impacted. All right, so that is how we go from DNA to a protein. I hope you found all this useful. Good luck studying.